Hey everyone, welcome to the third video in our 5-minute review video series. This time I'm going to focus on page 146 on the plasma membrane and the processes that occur because of the plasma membrane. So with the plasma membrane, it's very important that you understand how the structure of that plasma membrane is going to affect the function. So your phospholipid has those hydrophilic heads, hydrophobic tails, and it is a bilayer. And this bilayer in the center will prevent certain molecules from crossing. So anything that is large or charged, so large and in charge, it's going to have difficulty crossing due to that hydrophobic layer, and it's important that you remember that. So the plasma membrane is responsible for the selective permeability of the cell itself. It's going to allow objects in and out of the cell, or prevent objects from going in and out of the cell. And it's again all due to the structure of the bilayer, with the hydrophobic tails facing the interior and hydrophilic heads towards the exterior. So there's different types of transports, there's different functions for the plasma membrane. You have your passive transport, so passive transport is going to include diffusion and facilitated diffusion, osmosis. There's definitely, the key thing with this is that there is no energy involved, because substances are going to move from high concentration to low. And substances that move from high concentration to low do not need any additional energy in the form of ATP. Sometimes, such as in the case of facilitated diffusion, it will require a protein channel, or as is the case with osmosis, but there's still no additional energy that's involved in the process of moving substances across the membrane. Substances that are going to be able to move across the membrane include small, non-polar substances, gases such as oxygen, CO2, and then as long as it's got a protein channel, it is able to pass through the plasma membrane. For active transport, there are pumps such as the sodium-potassium pump, and with active transport, it does require ATP. And now I put just does ATP, but it does require ATP. Substances are going to go from low concentration to high concentration, so against their concentration gradient. The breakdown of ATP is going to make this possible. We talked about the sodium-potassium pump. 3 sodiums are going to leave the cell, 2 potassiums come in. Now that you guys have done the section on the nervous system, you'll see you know why that's important to help with the action potentials. Otherwise, the sodium and potassium gates are useless if the sodium is not able to diffuse down its concentration gradient. The other type of active transport is going to be bulk transport, exocytosis, which is removing things from the cell, endocytosis, which is bringing in things into the cell. And it's important, in addition to this, that you guys understand the concept of solute potential. This was the potato lab that we did at the during the first semester, and solute potential gives you an idea of which direction water is going to move. And with the solute potential, you do have the formula in your formula sheet, but please be careful to make this negative. The ionization constant will be given to you, the pressure constant will be given to you. You just have to remember that the big thing about the solute potential is, and what we did in our lab, was that when you are given a graph, let's zoom in a little here. When you are given a graph at 0 concentration, this is a negative change in percent weight. This is a positive change in percent weight. And then 0 is going to be the isotonic solution. So when you're graphing a solute potential, the key thing that you need to remember in the equation is given to you on the formula sheet, but the key thing that you got to remember is that the C in your concentration is the molar concentration, the C in the equation is the molar concentration in an isotonic solution. So the C value in the equation will be only where that your line of best fit crosses the zero axis. That is the isotonic molar concentration of your solute potential. I hope this video helps you guys, and good luck studying. If you have any questions, go back to your first semester notebook. We've got tons of information, tons of notes for you there. Good luck studying.